Hi, this is A Plus with the standards for Unit 8, Acids and Bases. If you look at this document, you will see after the main standards, you've got this pacing guide. The pacing guide will show you approximately the time frame for each type of lesson. If you've got a 50-minute lesson or a 90-minute lesson, there will be a review included and unit assessment. So the review is basically a teacher and student document where the students can review the concepts they have learned during this unit. So if you get time, try and cover every portion of the acids and bases unit. It will help them, those especially going into the advanced class of AP. So if we just look at the main things here which are involved with this unit 1, you have the definitions for acids and bases. So we must make sure fully understand. If you get a chance to do the Lewis acids and bases too, that will be helpful so they understand some of the concepts for later on. We need to be able to differentiate and know the properties of acids and bases, how they are similar, different, key terms and words, like corrosive for acids and caustic for bases. And you'll also see the term here sometimes for bases being used. This is when they are in solution form, alkalis. So if you see alkalis, that's related to the bases there, okay, in the solution form. Then the important thing is being able to identify the conjugate. So conjugate of an acid would be the conjugate base, and the conjugate of a base would be the conjugate acid. So they're opposites. Formulas that we need to really know about is pH calculations, pOH calculations, and also knowing clearly what the different titration curves look like, so they can identify whether it is a weak acid, weak base, or strong acid, strong base type of curve. And you will see there's a questions on the sample practice questions which will refer to some of these things. And generally what we've done here with the buffers is to know what buffers are and how they operate. Having said this, I have included a slightly higher level question in the buffers practice question, which will require some of your students who are more than capable of understanding what Ka is, the dissociation constant for weak acids and bases. So if it's a weak acid, it's going to be a Ka. If it's a base, it's going to be a Kb. And really, it's the dissociation between the products, concentration of the products over the concentration of the reactants. And remember, when you look at the products and reactants, you do not include solids, you do not include liquids. So anything which is a liquid or a solid, you would eliminate as being one of your products. So it's going to be all the products multiplied by each other and all the reactants multiplied by each other, depending on how many there are. And similarly for the bases, it's going to be the same type of equation. But in this case, it's going to be for the base, the products of the base reaction over the reactants. So if we now just continue looking at the remaining portion of each section, we'll be able to see that there will be included with this a screencast. The screencast is a link to this particular video. So if you forget about anything, the main concepts, you can come back and watch this. So concept 1, this all again links to what I've put in my PowerPoint. So concept 1 is also on the PowerPoint. So if you cover the PowerPoint thoroughly enough and use the practice questions, you'll find that you'll have covered all the important parts of this unit. And like I said, understand the acid-base terminology and the characteristics and definitions of each one of these. As the first bullet point says, make sure they know Arrhenius definition, where a proton is given up for an acid and hydroxide ion is given up for a base, and Bronsted-Lowry, which are the main sort of ones which tend to be in the AP as well, which refer to a proton donor and a proton acceptor. So, and if you get a chance, I'll put a little bit about the Lewis acids and bases, which will also be useful for those who end up doing AP chemistry, as long as they know something about it, as sometimes those types of things crop up and it's just good to know about them. Bullet point 2 is to clarify acid terms and alkali, like I mentioned before. It's a base which is soluble, and explain the properties clearly. Again, they are on the PowerPoint slides, how they taste, and the reactions with metals, things like that. So if we go to the next one, which is concept number 2, and then after that number 3, if we just go through these, I've tried to keep this basic and simple. So what I've done is I'll just put down all the main points. So when you talk about the pH scale, tend to use the Litmus Indicator as the very basic one initially to start off with. So you can see that the Litmus Indicator is the basic one, where you have turning red for acid, blue for base, and there's a little thing on the PowerPoint too which will help students understand that. And then there's more about the Universal Indicator, which has a range of colors and pH numbers associated with it. Try and get the students to understand what the pH scale is. It's a series of numbers. The lower the number, the more acidic. The higher the number, more basic. The lower the number indicates more hydrogen ions, and the higher the number, more hydroxide ions. You also need to be able to explain clearly what weak and strong acids and bases are. Remember, weak acids and bases do not dissociate completely, therefore you don't always get complete hydrogen ions being produced or hydroxide ions produced, whereas with strong acids you'll get almost all products. In other words, you'll get all hydrogen ions and hydroxide ions being produced. And again, just to get this concept across, you may want to use the ASIM Sponge Lab as a quick little demo to show how the sponge changes in different solutions. And this is an indicator to whether it's acidic or basic solution, or whether it's a neutral water solution. And this is a nice little demo just to get students engaged. Concept 3, Arrhenius, Arrhenius acid bases and Bronsted-Lowry definitions. Again, this may take a little bit more time, but be clear to clearly explain the definitions here of acids, bases, and also definitions of the Arrhenius, and be clear to understand what conjugates are and what the conjugate acid-base pairs are. And there's a portion on the PowerPoint which goes through this as well. So you can see that on the PowerPoint there are some of those checkpoint questions are built into the PowerPoint itself. So it's going to be relatively easy to follow and give the students as much practice as you possibly can. If we look at concept 4 is looking at strong acid, strong base, and pH calculations. Again, for the pH type of calculations, you must make sure that you have covered the important formulas that go with this. So for example, you need to know what pH stands for, the negative log 10 of the hydrogen. If it's POH, it's going to be the negative log 10 of the hydroxide, and calculate pH and POH. So we need to practice questions with that. Further down, you'll see there's some important formulas. pH plus POH equals 14. KW is the product of the hydrogen and hydroxide ions. pH, POH, we just discussed above. And how to get your hydrogen ion concentrations if you know your pH value. And similarly, how to get your hydroxide ions from the POH. So these formulas need to be learned and practiced. So calculations can be made a little bit easier. And sometimes there are 2 or 3 steps on these. So we need to be careful how we approach these so students have sufficient coverage and practice of these problems. Concept 5 and 6. So let's look at concept 5 and 6. Concept 5, differences between acids, bases, weak acids and bases. Again, we need to understand the emphasis of complete and partial dissociation. So if we look at complete dissociation, solid arrow, double-headed arrows will indicate partial, so weak acids. We need to be able to give good, clear examples of each so students know and maybe learn some of these. We also need to try and understand oxoacids. Now, you may have done this in nomenclature as well when you were naming acids. So this can also help understand those concepts. Looking at concept number 6, titration of strong acid, strong base. We need to really know how a titration is set up and what a titration is really. It's a way of discovering the concentration of an unknown acid or base by adding a known amount of acid or base to it. So you'll end up with certain values, volumes, and concentrations from which you can work out pH. And you can also do lab experiments which will show you titration curves being produced from which students can calculate out the concentration of the unknown acid or base. The important thing here again when you're doing this, students need to be aware of what the equivalence point is and the term endpoint. Equivalence means that there are equal moles of acid and base. The endpoint is just beyond the equivalence point when you've added sufficient amount, so it's no longer exactly equivalent, but it tells you there's a color change. If you use an indicator, that change has completely taken place. So again, the titration curves, if you go through the PowerPoint, there's an online lab that they can do, and there's also other ones suggested in the documents provided for you. So if you look through the other documents, not just the standards, you'll find there's a list of all possible. The pacing guide which is provided will have the labs that you need to try and complete wherever you can. The more labs, the better. If you look at 7 and 8, 7 and 8 is pH indicators. Try and show the students as many indicators as you can, so they will be familiar with names of indicators and their color changes, and maybe even do labs to show things change color. Even make an indicator and see how it changes color of solutions you may find at home. So that's quite simple and straightforward. And again, do not forget to link it with the pH that those solutions will be when they test them out. If they do this, they will remember quite a lot of the color changes. Concept number 8, which is all about buffers. Remember, we want to keep this simple. We just need to make sure that students understand what buffers are, where we find them, where they're used. So that way it'll be quite easy for them to understand why buffers are important and where in industry we would use these. For example, our blood is a buffer. They use buffer in lots of industries, pharmaceutical, where they make tablets. If it's not the right concentration of acid or base, it may break down early. So think about all the examples given to you in your PowerPoints. That will help you quite a lot with teaching this unit.